So I was going to gloss over this few slides real quick, but I realized, there's some of you probably would appreciate the history of rose rosette specifically. And it's not a new disease. It's been reported being present in the 1940s out of Canada. And I managed to get a screenshot of that document. And what's interesting about document is there's a total of less than, I think, 12 words and 2 lines describing a witch's broom. That was the first instance that at least is, by the way, I put 1940. It wasn't reported in paper in 1940. The publication was actually 1941. And then when you see this next one where it says 1941, that was actually a publication, I think, from 1950, looking backwards. So the dates here signify when it was observed. So 1941, Wyoming, 1942, California. Then it goes up 1960s, California. Still, oh, where did I go? Oh, right. Next slide. In the late 50s, when Nebraska at the North Platte Experiment Station on breeding stocks and on multiflora, 76, Kansas, in the 70s, Eastern Kansas, Western Missouri. That was the first instance, I think, was a record that basically noted that there were symptoms from hybrid roses essentially in urban or suburban type areas. This was the fun thing that I got out of that 68 paper, Ellington et al., which basically states it's unlikely, therefore, that rose rosette will be troublesome on cultivated roses in urban or metropolitan areas. Go figure. Moving forward still, 82 Eastern Oklahoma, 82 Northwest Arkansas, showing up there. If you think about the where the Mississippi River is, jump over to the eastern side of the Mississippi River in the mid 80s. So it shows up in Illinois, Kentucky, Indiana on naturalized stand of multiflora. So if you can kind of logically think, gee, guess how it got there? No comment on that one from me. Texas, it's showing up in the 90s. I'll put it this way. I think we got it from Oklahoma. And of course, then you also have a divergence then mid 90s where you see it popping up or reported in Tennessee, Ohio, Pennsylvania, West Virginia, 96 in Maryland. And then this is the fun part where we start getting into the nitty-gritty of the biology. In the 80s, paper by Gregorich and Kim. Oh, it's a double membrane bound virus-like particle. 87, there was demonstration. Hey, it looks and acts like a virus because we can inoculate it and we can graft inoculate it. And then in the 90s, oh, look, we still see stuff that looks like a virus-like genomic material in symptomatic multiflora rose. Then in 2011, out of the Arkansas group, that there was the virus that was characterized and named rose rosette virus. I don't remember why I put this. I think this was one of the discussions that I had with Jim Amrine at some point, where it's some of you may know this, some of you may not. At one point, rose rosette disease was thought to be a potential biocontrol agent to be used to reduce multiflora rose nuisance. So go figure. 2014 was when we started the SCRI Combating Rose Rosette project. So that would what I refer to as Rose Project #1. Real quick on the transmission, for those of you that might not realize this. The primary mode of transmission of this virus is believed to be the aerophyte mite Phyllocoptes fructiferus. It is able to acquire that virus when it feeds on a diseased plant, and it's able to transmit it when it moves to a new healthy plant and feeds on there. That's the belief. How does the mite move? Well, they could walk, so typically when you get rose bushes that are intertwined with each other, they'll go from one bush to the next. But more excitingly, this aerophyte mite can balloon and be carried by wind. So think paratroopers. This is special forces. I think Jan Olsen at one point actually had a video of them being taken up in the air. So that was pretty cool. Yes, sir. So the question is how do you explain to the general public the role of Rosa multiflora? I think you want to be real careful with that. One of the beliefs, at least among scientists, that the consensus is how it spread across the United States was probably by multiflora stock that was used for wildlife planting at some point, I think in the 50s and 60s. In the 30s, oh, the CCCs, through the CCC, I stand corrected. I am corrected by Dr. Windham. Thank you, Dr. Windham. I'm a lot younger than Dr. Windham. So in fact, the belief that the spread of this virus was probably true Rosa multiflora in the 1930s or so. So the host was there. Yeah. So multiflora is in fact a host. So by it being infected, we now know too that sometimes the symptoms are not always obvious when the plant is infected. So there's a very good possibility that it was moved around to places where it was used. So to your question, what is it? Well, it's a host. It was a host that was probably distributed by humans. Yes, sir, Dr. Hawkinson. So to those of you online, there's discussion there that Dr. Hawkinson is providing some explanation as to the nature of multiflora rose being aggressive and good growing. So along that lines, I actually tell a story, that I made up for the most part, where I said, this is smart because at some point somebody said, well, let's use rose rosette virus to kill multiflora. Well, you infect the multiflora rose, but I guess they didn't consult horticulturists because, boy, multiflora rose can put out a lot of seeds. And, that means those seeds, when they come up, those are potential sites for infection to occur. So it's only, helps it to get established in any given area. So that's a great discussion. I think that can continue further. But going back to the what I would call disease transmission process, as far as we know, and we still truly believe that all roses or almost all roses are susceptible to this virus. There's a point of argument that I have with Dr. Windham here on how fast it kills the roses. You have? Wow. So at one point, he was adamant that it would kill the rose within 2 years. And I told him, I said, in the Dallas-Fort Worth area, I've seen roses survive for 6 to 8 years before they totally go kaput. And but short of it is from what I understand, is this virus does impact the physiology of the plant itself, making it not a very good storage, very good in storing food during the winter. So it's susceptible to the cold. And I think that's what really kills it in terms of longevity. Yeah.
And you cannot, Sarah, it's almost impossible to transplant heavily infected roses and expect them to survive. Where if you transplant a healthy rose, it does fine.
So
Yeah, when we try to transplant them, I would say close to the 75.
So we're still learning a lot of stuff, and fact of the matter is there's still a large gap of information that we don't know. What time do you have? I don't have my watch on. Oh shoot, I gotta wind it up. Oh no, we're succumbing to Jean. We're taking over her time. So no, we talk about this. So real quick, I do want to point out one thing. So what I have here is really a chart on two things that were just drawn from the objectives of the project. So Rose Project 1, Rose Project 2, Project 1 being combating rose rosette that ended in 2019, Sustainable Rose Project in 2022 to 2026. What you see highlighted there is primarily where we see extension outputs and outcomes being involved in it. So in the first one was coming up with a best management practice, practices to combat rose rosette. That we do it well, kinda. Most of it thanks to Dr. Windham. Second thing is create an information and demo pipeline information hub through roserosettedisease.com. Well, that has morphed into roserosette.org. So that was the outcome of it. The last thing that was standing in that roserosette.org was that map and reporting thing. So I think after this meeting, I'll have Joe basically will shut that down and clear out the information. And then with further discussion, hopefully from here, we'll figure out what to do next with the maps and the data we'll still use for research. For the new project, the last thing that I highlighted there is where we have the extension group involved in primarily, which is develop comprehensive research, demonstration, and education program for rose rosette management. So I'm very thankful that several of you do partner with us to put out trials. So I'm thinking Matt and Jeff and the folks from Aramark, thank you very much to let us put things out there. And to point out, and I'm going to skip through a bunch of this. What time do I actually have to stop? Okay, what time does Jean has to stop? All right. So one of the things about, one of the things that I want to bring up is the whole thing about the research demonstration. So when this project was put together, the extension group got together and said, what can we do? Because we want to put out trials around the country and to be able to take data sets that are somewhat standardized so we can actually look at it in its entirety at the CONUS level, so on the continental United States level. And how do we encourage collaborators to join us in this effort? So folks, where we can put out trials. And a secondary component to that is how do we get citizens to join us in this effort, not only to create an awareness about the disease, but also to engage them in helping us gather information. So the three modules that the group came up with was a rose resistant trial, cultivar trial, a miticide trial, and a physical berry trial. So the concept of this is, well, their genetics right now, this is working with the breeders. There are varieties that appear to have resistance, or at least some level of resistance to rose rosette. We want to know what it is. We want to test it and see how it is in the landscape. The miticide trial, there is few and far between materials that are available for use. And for a homeowner, unfortunately, the only one that probably is readily available to them is bifenthrin. And so the concept is, can we put out trials at certain places to be able to evaluate that and look at the effectiveness of that to reduce incidence of rose rosette. The physical berry trial is mostly work that came out of Mark Windham's project, is by putting something in there to disrupt, I guess, the wind flow or airflow to use the barrier to capture or at least impede the movement of the aerophyte mites to where the roses are being planted. So the three things that you saw partially there were we actually wrote out how-to modules. The idea is to share this with potential collaborators who said, yeah, we're wanting to do this. And it lays out what we want to do and expectations on our part and expectations of what we want from them. And so that way, to try and promote for a more successful type project. So what we're going to talk about right now is I'm going to have some of our extension group folks, and maybe even if time permits, as some of collaborators just kind of share their early experience. Plans started going in, and in fact, for this year, we have only focused primarily on the rosette disease resistant cultivar trials. So that has been put in multiple places in the US. The miticide trial probably will be rolled in maybe next year or the year after, as well as the physical barrier trial. I will say this, the second and third one is a little bit more difficult to execute. One is with the miticides, you're dealing with chemistries. The physical tree with the physical barrier is a little bit harder because sometimes it has to deal with some construction or different types of plants that might we might have to rely on height and so on. So it's hard to standardize on that part. Yes. Yeah. So like I said, I'm hijacking this because in the afternoon, we are going to roll out how we're going to engage the citizen scientists in getting that information, because I realize we have some of our public garden collaborators here. And so we want to be able to show you how we're going to tag it, how we're going to utilize the data, but also the fact that that information is something that, or that system is something that the researchers as well as our collaborators can use to gather information and utilize that information. So that will be this afternoon, and hopefully Joe can cover that in 30 minutes. So going forward, let me just give a snapshot of what's happening in Texas. We did have one trial put out just about a month and a half ago in Baylor with 60 plus plants. And the idea is all those plants were going to go in this year, and we start collecting data in spring of next year. Couple of months ago, well, maybe three months ago, I don't remember. I had to take a bunch of plants up because I screwed up to Matthew and Mobot. So a long van drive with a couple of hours, and I'll be itching to see how that turns out. So those are the two things that I've been involved in putting in. And in fact, we're putting in a third trial in the Grapevine area, and that's pulling out a trial that has been there for about 5 years with other varieties.